So as always, making sure to record our meeting here tonight so that if you ever want to go back and see what we talked about and watch me go over it again, you are more than welcome to. So for tonight, we are jumping in. Oh, one thing that I did want to bring up before we go ahead and get started is that next week is the last week of classes we have before the Thanksgiving break. So we have one more week of class and then 1 week of break before we get into the final push of the school year, which is nice to have that break. The downside of it is that next week is also when our first exam for this class, our first test, will be posted. So I'll talk about it on Tuesday when it gets closer. The exam will go up on Wednesday morning and we'll cover everything that we've talked about in this class so far. So the DNA, the replication, all of the polymerases and the helicases, the protein synthesis that we're talking about this week, as well as meiosis and mitosis, the subject we're going to be talking about next week. All of that is going to be covered on the exam. So because there's so much information on there, it is going to be a little bit longer than your normal homework. So even though the exam is going up Wednesday morning, it is not due until the end of the night on Friday. So you have all day Wednesday, all day Thursday, and all day Friday to work on the exam. Like I said, I'll talk more about it on Tuesday, the day before the exam goes live. But I wanted to make you guys aware of it now, just so that you're not sort of caught off guard by the time that we get there next week. And then the nice thing about it is that once you're finished with the exam, that'll be the last work that you have to do for a whole week, and you can sit back and enjoy your Thanksgiving break from school. So that is coming up next week. But for this week, we are finishing up talking about protein synthesis, so making these proteins. All right, so let's see, where is a good kind of reviewing point? So last week we were talking about how all of that information about our body, all of our genes is in our DNA, and that those genes are used to make proteins, and the proteins go out and do sort of the work in our cells. So this whole process is called protein synthesis. Having this DNA template and using it to make a protein. So yesterday we talked about how we start off with the DNA and then we copy it into a new strand, a new substance. Does anybody remember the name of that single strand that the DNA gets copied onto?
Is that RNA?
It is RNA. Specifically, do you remember what kind of RNA?
It's like a ribosome.
Not quite. The ribosome is actually what makes the protein. But the RNA that does it is the mRNA. Yeah, Paolo's got it. So we start off with our DNA, and then we have to copy it onto a string of RNA, specifically the mRNA. So we'll go back to those in a second. But specifically mRNA, which is what copies the DNA code and takes it the ribosomes. So does anybody remember why is it that we need this mRNA? Why can't the ribosome just read the DNA right off the bat? Why do we need this messenger in between?
What was it?
So I'll give you all a hint. Does anybody remember where the DNA is in the cell?
The nucleus.
Bingo. And is the ribosome in the nucleus?
Yeah.
Not correct. The ribosome is not in the nucleus. And that's where the problem is. So our DNA is in the nucleus. The ribosome is not in the nucleus. The DNA can't leave the nucleus. The ribosome is too big to go into the nucleus. So we need this mRNA, we need this messenger RNA to copy the DNA code and carry it out of the nucleus to the ribosome. So that's why it's the messenger. That's why it's the copier. It copies the DNA and takes that genetic information, takes that sequence of A's and C's and G's and U's over to the ribosomes. So some of the other main things that we talked about on Tuesday was that RNA is different from DNA in a couple key ways. It has a different sugar, ribose sugar versus deoxyribose. It has a slightly different set of bases. So DNA has the base thymine, RNA does not. It has the base uracil. So in RNA, anywhere where you would normally put a T, instead you put a U. And then RNA is single-stranded while DNA is double-stranded. So DNA has this double-stranded helix form going around. RNA is just the single-stranded like ribbon kind of structure. And then again, DNA has thymine. RNA replaces that thymine with uracil. So no T's in RNA, U instead. So if you see something that has a T in it, it cannot be RNA. It has to be a piece of DNA. Same thing with U. If you see something that has a U in it, it cannot be DNA. It has to be a piece of RNA. So different sugar, U instead of T, single-stranded versus double-stranded. So the main step of this process that we talked about last week is we open up our DNA and we build out our little mRNA strand by matching the bases. It's just like we did when we were copying our DNA, only now, instead of hanging around, the mRNA is going to leave from the DNA. And also, an A on the DNA side matches up with a U on the RNA side. Because remember, no T's in RNA. We have U instead. Everything else still matches the same way. A T in the DNA matches with mRNA's A C matches with G matches with C, so on and so forth. The only difference is that an A on the DNA side does not match with a T because RNA doesn't have T's. It matches with a U instead. So this is sort of the main first step of this process. It's called transcription. And it's where we take DNA and turn it into mRNA. So now we have our mRNA strand. It's going to head out from the nucleus. So it's hanging out in here in the nucleus. It makes this copy of the DNA, and then it pieces out. It's got to leave so it can go over to the ribosome, which is like our little protein factory. So the way that the mRNA is set up is that it's made up of multiple sets of what are called codons. And codons are a 3-base code in either the original DNA or more normally the mRNA strand. So 3 bases in a row make up one codon. So like here we have AUG is a codon. GUC is another codon. CCA is another codon. UAA is another codon. So the way that we create a protein in the ribosome using this code is that amino acids are the building blocks of our proteins, right? If you remember from way back in the notes, if we take a whole bunch of amino acids, put them together in a chain, and then fold that chain all up in a specific way, we get a protein. So our amino acids are our building blocks, and our protein is our final structure. So these codons that are in our mRNA, these series of 3 bases, code for one specific amino acid. So, for example, the codon AUG is the code for methionine, this like whitish amino acid. A lot of times you'll see amino acids represented as kind of like these balls or these bubbles. So that's what you might see in some of the pictures on the slides here. So AUG is the code to make methionine. GUC is the code to make valine. AGU is the code to make serine. And CCA is the code to make proline. So each one of these amino acids is coded for by one codon, one set of 3 bases. Some codons don't code for amino acids. Instead, they are known as stop codons because they tell the ribosome that this is where you need to stop adding amino acids. So like in the case of this really, really short little sequence, UAA isn't a amino acid codon, it is a stop codon. So once the ribosome reaches the UAA section, it knows that we're not making any more amino acids. We are done putting this protein together. So 3 bases are a codon, and codons tell the ribosome which amino acid to add next. All right, everybody okay with this so far? Any questions about the codons or the amino acids right now? All righty. So how exactly does the ribosome read this RNA sequence in order to put the amino acid with the correct codon? It gets help from another type of RNA called tRNA. So mRNA is our big, long, kind of single string ribbon that carries the message from the DNA to the ribosome. tRNA is kind of funky cloverleaf structure. So I'll show you a picture of it really quick. So it's this kind of funky clover, or I think it sometimes looks like an old-timey key kind of thing. And it is a different type of RNA. So it still follows all the same rules. It has uracil. It's just a single strand. And it has that ribose sugar in it. But the tRNA has 2 important sections on it. So one end of it is the attachment site for a specific amino acid. So each tRNA has a certain amino acid that is attached to it. So these are sort of like the taxis or like the Ubers for the amino acid. They carry the amino acid around to wherever it needs to go, specifically to where it needs to go in the ribosome. So one end is where the amino acid gets hooked up. The other end has a sequence of 3 specific bases. These ones are not called codons. In tRNA, its bases are called anti-codons. The reason for that, and I'll show you a picture of them at work in a second, is that if we have our 3 codons in our mRNA that are going through the ribosome, the anti-codons of the tRNA are what will match up to make sure that we've got the correct sequence, the correct amino acid. So if our mRNA had a sequence of CCC, our tRNA would have an anticodon of GGG. So then as the mRNA is going through the ribosome, the tRNA comes in, matches up, makes sure that it's the right one, and then will essentially drop off its amino acid to the next part in line. So like I said, I'll show you kind of a picture of what that looks like in a second. But again, here's our basic tRNA structure. Again, on one end, so up here is where the amino acid is going to attach. So this is an empty tRNA right now. So it'll go out into the cell, pick up a specific amino acid, which will attach right around here, and then carry that amino acid back to the ribosome. Once it gets to the ribosome, the anti-codon of the tRNA will match up with the codon of the mRNA So they match up together to make sure they've got the right one. And then it'll let go of its amino acid, drop it off, and head out back into the cell to pick up another amino acid. So here's sort of a little bit better diagram of how this works. We have our mRNA strand. It's at the ribosome. It's going through. And the ribosome is reading each codon that comes through. So we finished doing these three already. Now we're on to the next three. In this case, our codon on the mRNA is ACU. That means that we have to match up in order to get the correct amino acid down here. We have to match up with the correct anticodon on a tRNA. So if our codon is ACU, then our anticodon has to be UGA, because the A of the mRNA will match with a U in the tRNA, C in mRNA matches with G, and the U in the mRNA matches with A. So once the anticodon and the codon match up with each other, this amino acid will come off of the tRNA and attach to the big string of amino acids that we've been building. This tRNA will leave. It'll head out to pick up another amino acid, and the ribosome will move on to read the next codon. So the next set of 3 bases will be read. The correct tRNA will come in and it'll drop off its amino acid, so on and so forth until all of the correct amino acids are put in sequence and we reach one of those stop codons that tells the ribosome not to read anymore. So like I was saying, each codon is a code for a specific amino acid. So each set of 3, so ACU, GGG, CAC, CCG, all of those are specific amino acids. However, if you do some quick math, which I know it's late, so you probably didn't, and you had to remember from yesterday, I mentioned there are 20 different amino acids that we use as humans, but there are 64 possible codons. So each letter could be one of 4 different bases. So 4 times 4 times 4 is 64 possibilities. So a lot of times, multiple codons will code for the same amino acid. So usually, a lot of times it will be like CCG, CCC, CCA, and CCU all code for the same amino acid. So sometimes we get duplicates or kind of copies of the codons for the same one. And like I mentioned, sometimes a couple of them are stop codons. So the stop codons don't code for an amino acid. They just tell the ribosome to stop and not make any more amino acids. So early on, when they were discovering the mRNA and the codons, scientists pretty quickly put together a chart or a list of which codons code for which specific amino acid. So they ended up with a chart which sometimes can look a couple different ways. This is a little bit kind of nicer version of the chart. And on this chart, you read the codon code by starting with the first letter, which is the big letters in the middle, and then the middle letter is the smaller letters in the middle ring, and the third letter is the really small letters in the outer ring. So, for example, if your codon was AUG, on this particular chart, you would start by looking at the first letter is the innermost part here. So in AUG, the first letter is A. So you'd start down here. Next letter is U. So you look in the A section for a U. Here's the U. And then after U comes G. So you look in the U section, AUU, no, AUC, AUA, AUG right here. And then in really small letters down here, you can see that AUG gives us methionine. So this is a really nice version of the genetic code chart that's fairly easy to read. Unfortunately, a lot of scientists, and specifically the biology STAR exam, do not use this version of the genetic code chart. They use a version like this, which looks a lot less pretty, but has all the same essential ingredients. Instead of looking from inner to outer circles, on this chart, you start with the first letter over here on the left, and then you find the second letter across the top, and the third letter down the right-hand side. So in this chart, for example, you can see that A, if we wanted to do AUG again, our first position is A, so we look for the A box. All right, here it is. Our second position is U, so we look for the U box. Okay, the first one is U. So A, U, and then we can either look all the way over at the right, or they have them all listed here in this section. So A, first letter, U, second letter, and then we look and find AUG is methionine, or abbreviated as MET. So that is how you read a codon chart. You can figure out for each codon, each 3-letter sequence of your mRNA, you can know which amino acid is going to be put in that place. All right, so we're going to do some examples together where I want you guys to help me figure out which amino acid or which codon goes with which amino acid. So let's start off with this first one. If we have the codon GGG, what amino acid are we going to get?
GLY.
GLY. Perfect, Paula and Roberto. So G is our first letter. So we know we're going to be somewhere in this bottom row. So we go to the top for the second letter. G is our second letter. So we're going to be in the bottom row, last column, which is over here. And then like I was saying earlier, a lot of amino acids have multiple different codons that code for the same thing. So GGG codes for glycine, abbreviated as gly here, which is correct. And then, but also GGA, GGC, and GGU, they all code for the same thing. let's look at the next one, UCA. What codon does UCA, what amino acid does the codon UCA represent?
SER.
SER, serine. U, right here, so top row, C, right here, second column, UCA, serine. All right, CAU, what are we going to get with CAU?
HIS.
Bingo. C, first position, so we're in the second row. A, second position, so we're in the third column. So we know we're going to be somewhere in here, and then yeah, CAU gives us histine, or HIS. All right, GCA, what is GCA going to give us?
ALA.
Exactly. ALA, which is short for alanine. So yeah, G right here, C right here, and then anything that starts with GC is automatically going to be alanine, including GCA. And then finally, what about AAA? They're a great car maintenance program, but what type of codon or what type of amino acid is AAA going to result in?
LYS.
Yep, LYS, lysine, right here. So yeah, A is first, A is second, and so AAA is going to give us lysine. So you guys have just taken a series of seemingly random letters on an mRNA strand and turned them into a sequence of amino acid that is going to be turned into a protein. So is a chart that I literally pulled straight from the biology star test. So if you have to take the biology star at some point in the future, and you see this codon chart, you now know how to read it in order to turn codons into amino acids. So like I mentioned, most of these codons, a lot of them code for the same amino acid. And then a couple of them, especially in the U's, so UAA, UAG, and UGA are stop codons that tell the ribosome, no more amino acids, we're all done. All right, so does anybody have any question about the process of turning mRNA strand into amino acids using the ribosome and using those little tRNAs that are coming in and out?
I'm good.
All right. So this process, this final process of taking the mRNA and turning it into a protein is called translation. So here's sort of a super duper basic overview of what we've been talking about the last couple days. So we have our DNA strand, and then that's inside the nucleus. So this purple part of the cell is the nucleus. We're going to transcribe this DNA and copy it into an mRNA strand. This mRNA strand is then going to leave the nucleus and go through the ribosome. As it goes through the ribosome, those tRNAs are going to come in and one codon by one codon match up the mRNA codon with the tRNA. What's it called? What's that lower part of the tRNA called that matches up with the codon?
the mRNA?
Well, the codon is on the mRNA. And then what's the part on the tRNA called that matches up with the codon?
Well,
It's like the opposite codon.
Is it the anticodon?
The anticodon. Bingo. So our mRNA has the codon. The tRNA has the anticodon. Those match up together. And when they do, we take that specific amino acid that the codon is coding for, whether that's an AUG to make methionine or a GGG to make glycine. And we drop off that amino acid. And so this little chain of dots right down here is all of the amino acids being connected together that will eventually form a protein. So here's kind of another, a little bit more broken down version of the process. So we have our big long strand of DNA. Different parts of that DNA are different genes. So in order to get each gene to be turned into a protein, we first open up and copy the DNA into an mRNA, where again, we don't have any T's in mRNA. We change those T's to U's. So the A's match with U's, the C's match with G's, the G's match with C's, and the T's match with A's. So we go through transcription and turn DNA into RNA. That RNA then heads out from the nucleus, goes into the ribosome, and each of these codons matches up with the tRNA anticodon and codes for a specific protein. And that's called translation. So this happens, the translation happens inside of the ribosome. The transcription happens inside of the nucleus. Because remember, the DNA is stuck in the nucleus and the ribosome is stuck outside in the cytoplasm, the like regular part of the cell. So we need to make this mRNA inside the nucleus and then move it outside into the ribosome in order for this gene to get turned into a protein. So whenever you hear somebody talking about your genes and what they do, like on the news or on a science show, at this very basic level, all that your genes do is code for a certain protein. And then it's the different proteins in your cells and in your body that perform all the different functions that determine how you work and how you use energy, take down energy, and use it in order to grow and survive. This picture I actually like kind of the most out of all of them because it gives a really good and kind of really breakdown overview. So again, we have this DNA inside of the nucleus. When we want to turn a gene into a protein, we've got to open up the DNA and copy that gene, turn it into an mRNA through transcription. That mRNA is then able to leave the nucleus, come out into the rest of the cell, head on over to a ribosome, where each set of 3 bases, each codon, matches up with its specified tRNA, and then the tRNAs all drop off those amino acids to kind of this growing chain. So the tRNA basically comes in, locks with the mRNA, those codons and anticodons lock together. the amino acid moves from the tRNA to the chain, and then the empty tRNA leaves to go pick up a new amino acid. So you can see these guys are coming in with their amino acids on board. Then these guys are leaving empty to go get new amino acids. All right, so the last thing that I wanted to show you is that video. We started watching it yesterday when it went through just the transcription part, but now I want to show you the whole thing because I think it does a really great job of illustrating, especially all of the moving parts that are going on in our ribosome down here. So let me make sure. Can you guys see the YouTube video that's loading up that has the big purple dot in it?
Yes.
Yes.
Okay, good.
Yes.
Always
Yep.
use to double check.
Mhm.
All right, so this is our cell. So it's the outer part of our cell where all this exciting stuff is happening. And then when we go into our cell, there are all these different little structures of the cell that I think you go over in more detail in biology A. But the main one we're going to focus on is the nucleus, this purpley part right here in the middle. That's what we're going to look at to start off with. Again, even though you can't really see them yet, but the ribosomes are on the outside of the nucleus. The DNA is all inside the nucleus. So we're going to go inside the nucleus to check out our DNA. DNA isn't just floating around all willy-nilly in there. It's packaged up in a certain way that we're going to talk about in a later week, so don't worry about that quite yet. But we do have specific genes in our DNA, and we have to turn these genes somehow into proteins. First thing that we got to do, go through transcription and make some mRNA. So you may remember we had DNA polymerase when we were talking about DNA replication, and the DNA polymerase put down all those new DNA segments. Well, in transcription, we have RNA polymerase that comes through and makes this big long strand of mRNA. So it's going through and matching up mRNA bases with the DNA bases. So if there's a T in the DNA, we're going to have an A in the mRNA. If there's an A in the DNA, no T's in mRNA, T's not allowed. Instead, we have U in the mRNA. C's still match with G's, and G's still match with C's. So we're going through, matching up all these bases. And then as we go, the mRNA, as soon as the bases get matched up and attached to each other, it starts to kind of peel off of the DNA because it doesn't really need to hang around. It just needs to make sure that this base matches with this base. Once it does and gets connected to the bases around it, then it can start to leave. So we're going through matching up base by base by base by base to copy this gene in our DNA into a strand of messenger RNA, also known as mRNA. Don't worry about this part. The mRNA just does some extra stuff after it leaves the gene, but that's not really important. Then the RNA is able to leave the nucleus, so remember this big purple ball is nucleus, and it has these tiny little holes in it called pores, just like the pores on your body. The nucleus has pores too, which allow the mRNA to leave. So again, the DNA has to stay in the nucleus because it's too important. If it gets damaged, then your whole cell is screwed. So the DNA stays nice and safe and protected in the nucleus. The ribosomes are too big to come into the nucleus and read the DNA directly. They can't fit through these tiny little nuclear pores right here. So that's what we need our mRNA for. We can make the mRNA strand inside of the nucleus, and then it's small enough to be able to leave the nucleus and go out to the ribosome. So our ribosomes are our protein factories, and they come and kind of clamp down on the mRNA. And then here is the next star of our show, the tRNA. So it's got this funky clover key shape. We have our amino acid up here attached to the one end of the tRNA, and our anti-codon that's going to match up with the mRNA down here on the bottom. So as the mRNA goes through the ribosome is essentially coordinating the tRNAs coming in, matching up their anti-codon with the codon of the mRNA. And then as soon as one matches down here on the bottom, it drops off its amino acid and another one flies in and matches. So you'll see, I know it's kind of hard for you guys to see because Google Meet doesn't do a great job of showing videos. I'll do my best to remember to put a link to this video in the Google Classroom. So if you want to go back and watch it, it's only like 2.5 minutes long. And this part right here is really beautifully done to show this process. So what you'll see is that an mRNA flies in, matches up, or excuse me, the tRNA flies in, matches up with the codon, drops off its amino acid to this growing chain up here. And then once it drops off and its amino acid is empty, it flies right out to go get a new amino acid. So it comes in, matches up, attaches, flies out. And that's how we're able to build this big, long amino acid chain out of the ribosome. And eventually, once we hit one of those stop codons, the amino acid chain folds up and gets turned into a protein. So we have the gene in our DNA. It gets transcribed into mRNA, which can leave the nucleus and go to the ribosome. Every 3 bases on the mRNA, every codon, matches up with a tRNA carrying a specific amino acid. And so as the ribosome travels along the mRNA, it matches up those codons, puts the specific amino acid in, and grows a longer and longer chain that eventually will get turned into a protein. So transcription, copying the DNA into mRNA, translation, reading the mRNA and turning it into an amino acid chain, which will become a protein. right. Have any final questions about tRNAs, codons, mRNAs, amino acids, anything like that?
No.
All right. So this is honestly one of my favorite things to teach about, just because I love that everything is this big long sequence of events where everything kind of fits together, right? So just by knowing a DNA strand, you can figure out what the mRNA strand is going to be, and then you can figure out from there, based on those codons, what the correct amino acid sequence should look like. So that last part, turning the RNA into an amino acid sequence, is actually what your homework for the Thursday assignment is based on. And so I give you this big familiar looking codon chart that we were just doing some work with a little bit ago. And I want you to tell me for each one of these codons, what is the correct amino acid that'll be put there. You could just put the 3-letter abbreviation if you want. You do not have to go and look up the actual full name of the amino acid if you don't do. But there's a series of, I think, 6 or 7 amino acid codons. And so for each codon, I want you to tell me which amino acid matches up with it based on our chart right here. So that is the assignment for tonight. Or since you guys are here in the Google Meet call, you already got your attendance credit. So if you want to work on this on Friday or over the weekend, that is fine as well. And then Monday, when we come back, the attendance question has to do with tRNA and kind of what it does in this whole big protein synthesis activity. And so that'll be your kind of Monday short little attendance question before we get into our next set of notes next Tuesday. All right. So does anybody have any final questions about transcription, translation, homework, stuff coming up in class, any final thing that you have any questions on?
No.
No. No.
No.
Thank you.
All righty then, guys. Well, as always, if you do think up a question